If you look at a wheat grain, but it's also for rice or for maize, in the middle there is this sort of white starchy bit, which is actually 70% of the grain. And so that's the starch, that's where the calories are, and that's what we like to eat. And just that there is not much iron in there, in any of these cereal grains. The iron is just very low. However, we've now shown that we can do it with, it is GM, but it's actually, it's only using the wheat genetic material. The genes are already there in wheat. All we do is, bring them in a different tissue where they normally, they're even there, they're just not activated in that tissue where we want them. We had on a computer screen, as it were, we had the genome sequence of wheat. And then you find the sequence that makes the iron transporter. So then there is a way of isolating just that, which is it's a tiny bit of the whole wheat genome, but you isolate it and actually you put it in a bacterium that will amplify it for you. There is a bit of a gene that encodes a protein, in this case a transporter, that literally takes iron from outside of the cell to inside the cell, or inside a cell compartment in this case. And in front of that bit there is another bit of DNA that regulates the expression levels. So what we've done in this case is taken the bit that encodes the transporter with a regulatory bit that's very highly expressed in the center of the grain. And so that's the change we've made. So these bits together, they then go into another bacterium called Agrobacterium. And that bacterium has actually, it's been around, for as long as evolution exists. It goes into plants and puts a certain bit of its DNA into the plant. And that is what we're using. So we basically use Agrobacterium to help us as a vehicle to put a bit of DNA in.